I have two different species of planaria. One have a round head and one have a flat head. They have some stem cells, and each planarian, when you cut off the head, they build a head of the correct shape, and then they stop. That's what planaria do. Now I have a thought experiment. I'm going to take my round-headed planaria. I'm going to take out some stem cells, and I'm going to inject them into the body of the flathead planaria. I'm going to let them get comfortable and move around and settle in, and then I'm going to cut the head off. What head shape are we going to get?
Hi everyone, welcome to Reason with Science. I'm your host Jitendra. This episode is with Michael Levin. He is a professor in the biology department at Tufts University. His work on left-right asymmetric body structures is on Nature's list of 100 milestones of developmental biology of the century. Here we talk about life as an information, deterministic view of life, multi-cellularity, two-headed flatworms, Picasso frogs, bioelectricity, and collective intelligence of cells. Enjoy the conversation, share, and subscribe to support the podcast. Thank you for listening. Hi, Mike. Welcome to the podcast.
Thanks so much. Great to be here.
Yeah, so let's start with the kind of your basic description of the work. Because the topics that we are going into, like afterwards, that they are really broad, and basically they somehow connect with your work, which is on morphogenetics and bioelectricity. So what is the best way to describe what you do?
Well, fundamentally, everything in my group and basically my whole career has been around trying to understand how different kinds of minds are embodied in the physical world. And in particular, we're interested in this idea of scaling up. So how is it that small competent parts come together to form some kind of a larger scale emergent intelligence? And the bioelectrics come into it because if you think about how it is that cellular collectives solve problems, how do cells make decisions? This is a really good way to start on this problem, and it's something that's been realized actually by some people for a long time. So, for example, Alan Turing was one of the forefathers of artificial intelligence. He was interested in mind in the physical world, but he also had this amazing paper on morphogenesis. Yeah, he's had one of the early papers on computationally mathematically modeling morphogenesis. And so you might wonder why would somebody who's interested in computation and mind be interested in morphogenesis? And I think he saw clearly that this is actually the same problem. And I would also say that each one of us makes the journey from just being a piece of physics and chemistry, so an oocyte, an unfertilized oocyte, and eventually 9 months and some years later, we become kind of a sentient human with all kinds of second order cognition and complex mental life and so on. And so we all traverse that journey from physics to mind, and understanding how that happens during embryonic development. What is it that allows individual cells to start off doing very simple kinds of things, and then solving problems in morphological space, and then becoming a kind of complex cognitive agent. That's what my group is really interested in.
Yeah, it's fascinating, and how important these questions are for the understanding of biology. And especially why I wanted to start with the brief introduction of your work, because then the next question that I have is simply, what is life, where the whole biology starts, basically. So how do you look at it?
To be honest, I don't think about the question of what is life at all. I'm not sure that life is a particularly useful category for us. People argue about it, and there are some people who do nice work on this topic. For us, I'm not sure it's very important. I think what is really important is to think about what is cognition, and what is it to be an agent, what is agency, how to define this sort of continuum of agency that has physics on one end and simple physical systems, and then has complex beings like us and beyond on the other end. I'm not sure the actual notion of life is of any help here. And I think that there are many cognitive systems that are not what we would call alive. And I don't think it matters really for us.
So at least for the progress of science, you don't think that this question is as important as, for example, asking the question of cognition or intelligence?
I think so, because fundamentally, look, this all boils down to one thing. Why do we care what the cognitive level of some system is, right? And I'm interested in systems that are very diverse. So the kinds of frameworks that I deal with have to deal with standard evolved animals like we see on Earth. They have to deal with all kinds of unusual living things that are evolved, swarms and colonies and cells and tissues and plants and all kinds of unusual things. Potentially exobiological agents, so anything we find off planet at some point, probably we will, and of course synthetic kinds of forms that we make here. Now those might be out of living material, those might be out of traditional AI, so software and some kind of a silicon medium underneath, and of course chimeric hybrid constructs, so biorobotics and cyborgs and hybrids and all of those things. So frameworks have to be able to deal with all of that. And so why are we doing all this? We are interested in these questions because we need to know how to relate to these things, right? At one end, you're talking about how do we relate to them from the perspective of control. So maybe engineering, maybe robotics, maybe regenerative medicine. How do we control the outcome of these collective intelligences? On the other end, you're dealing with things like social policies. How do we, when we do have all kinds of novel beings among us, which we are going to have, that are partly standard human, partly different kinds of tissues, partly embedded smart implants with AI and all these kinds of things, and potential alien intelligences. How do we relate to them, right? And asking the question of whether they are alive or not seems to me irrelevant to that question, but asking what kind of cognition they have seems very relevant. So to me, it's all a question of what is the correct way to relate to other beings around you of various degrees of sophistication.
Yeah, that's fascinating, because it also, I mean, to start with, first of all, this information we get from our biology textbooks. This is how things start, that okay, that in this hot springs, in this hot soup, the life started, and et cetera, et cetera. So also kind of to see now where we stand on these kind of questions. The next point is, I mean, of course, there was a lot of information that you already gave, basically the gist of different topics that we'll be covering in the conversation. But let's start with as you said, the computation part. But before that, life as an information itself, because let's say that, okay, I mean, even if we kind of avoid or remove the question of life part, but still life exists. I mean, there are systems that we consider as living beings, but I don't know, even if we remove the semantics part, the point being that there is certain information, there is some blueprint that we all kind of follow, right? So where do we stand with the life as an information part?
Well, let's look at the semantics issue, because it's kind of important. Let's just decide. There are two types of terminology that we can use. There's terminology that are useful in everyday life. So for example, the word adult is like that. So we all know what an adult is. It's very useful in court. It's very useful when you need to rent a car. This is very important. But we also know that scientifically, absolutely nothing happens on your 18th birthday that makes you a different kind of creature that now you can do other things, right? It's total fabrication. So we need to be clear. Are we talking about terminology that's kind of a heuristic that's useful for life, for society and everything to move along, or are we talking about deep scientific concepts? So now there are people who do very good work in this area. So Sarah Walker, Lee Cronin, those folks really think very hard about what life is. It's great. For the kinds of things that I'm interested in, I don't believe that a binary category of life versus not life is very useful at all. And in fact, I don't believe almost any binary categories are useful, because what you end up with is these sort of semi-philosophical questions about, well, how about these new viruses with a little bit of their own physiology? Are they alive? I find this completely sterile for the kinds of purposes that we have, which just as sterile as questions of which sometimes people talk about, whether something is or is not cognitive or has agency. I think that's a terrible way to frame these problems. The question isn't, is it or isn't it? The question is, what kind and how much? So I have this very continuous view of these things. I think that what I really want to know when you show me a new system, and it may be a surprising thing that you either made or you discovered or it came off planet or whatever it is, you show me a new system, I'm not looking for you to put it in binary categories. I want to know what I can expect. Can I expect this thing to only follow immediate energy gradients like a rock or a bowling ball? Or can I expect it to act like a homeostat and keep to certain parameters? Can I expect it to be trainable, like with rewards and punishments? Can I expect it to give me wisdom, and so we can have a an equal relationship with it? Or can I expect it to be so much more advanced that whatever, we can have a different relationship with it? So to me, all of these claims are relationship claims or engineering claims. It's all about, what does it enable me to do? How does it enable me to optimally relate to this? And I think asking whether something alive or not on an everyday basis, of course, that's very useful. On a scientific basis, I'm not sure it does very much for us.
Yeah, the only thing is probably it may just explain the fact that there is a transition, and if there are these kind of basic laws which do explain transitioning from chemistry to biology, because most of your work, I mean, that's what we will discuss today as well. Because your observations, once we start expanding, somehow they go to also kind of chemical level, right?
Yeah, I mean, this whole issue of transitions, maybe there are transitions, but I think that if you want to argue that there are sharp transitions, meaning some kind of phase transition where something very radically different happens, that may be, but I think then one has to argue for that. Well, what is it? What is it? What exactly happens? And I'm sure there are phase transitions, but I'm not aware of any that are kind of defensibly sharp. They tend to be, in my mind, they tend to be very slow and gradual, and I mean, there certainly are new kinds of capacities that arise as one goes up certain levels. But I've gotten more use out of focusing on the continuous nature than focusing on kind of the trying to put things in categories. Actually.
No, that's definitely true. I think there is no, or at least I'm not aware of any biological system which is like this phase transitioning. I think all of them, they are gradient, right? I mean, of course, then there can be some selection or something that may work, which may remove parts of it, but mostly it's the gradient switches, right?
I think so. A lot of people don't agree with that. A lot of people think that there are very sharp boundaries. So there are many people that think that certain kinds of, for example, certain kinds of animals have agency and cognition, and others do not, and that's it. And there's a sharp line. And I mean, I've never heard of a plausible sharp line from anybody, but there's certainly plenty of people who believe that, and even sort of on the lower end. So there's some very nice work on minimal agential systems. So those little kind of very, very simple chemical systems that show interesting behaviors. I don't think people would call them alive, but I spend zero time asking whether they're alive. I want to know, what does it do, and how much, where does it fit on this kind of continuum of agency? Droplets and active matter and those kinds of things. Yeah, I don't know. I'm not terribly interested in whether they're alive, but I do want to know. So what can they do? Do they learn? Do they pursue goals? What kind of goals? How large are their goals? That kind of thing.
Actually, I interviewed a chemist, Adi Pross, and he has written a book like this, transitioning of chemistry to biology. And this is what his definition was, that living systems are self-sustaining continuum systems. So, like in a way that, because what you are saying, that once we start extending, I think that's what, even at the chemical level, will end up with the similar kind of understanding.
Yeah. I mean, people certainly do come up with definitions. around that, and then somebody will say, yeah, but fire does that too, and they say, no, you need heredity. And so there are some, there's some categories. I'm not sure. I think a lot of those distinctions have to do more with the question of what department gets to study them, as opposed to what the real phenomenon is. So a lot of these things that are studied in chemistry, in biology, in computer science, in psychology, those barriers between these departments are getting weaker every day. All the important, interesting problems are multidisciplinary problems that don't fit neatly into any of these categories.
And what do you mean by the continuum system in biology in that case? So for example, if we take viruses or like those organisms, how would you look at them?
So I've proposed, and I'm not the only person to propose such a thing. Of course, people have done this before, a continuum of cognition. This continuum is defined by different types of different capabilities with respect to how you control it. And I can describe what the continuum is. But the point is that it's a very sort of functionalist cybernetic kind of definition. It's very engineering based. It does not care at all what the system is made of or its origin story. So whether you were designed, whether you were evolved, whether you came from a factory or you evolved naturally on some planet somewhere, or what you're made of makes no difference for the purposes of this kind of framework. What matters is what kind of cognition you have. So along the continuum, as I was saying before, you might have, and it's all really about, if I'm an engineer or some other observer and I want to interact with the system, what is the most efficient way for me to do that? So it might be a system that's way on the left of the continuum. It might be like a mechanical clock. So it's a piece of physics. If you want to change what it does, you can rewire it. You can physically change the hardware, but that's it. You cannot train it. You cannot change any set points. You cannot argue with it. It makes no sense to be angry at it. It's a piece of physics. That's it. Then, sort of somewhat to the right of that, and I'm sure there are many categories. I'll only mention a few. To the right of that, you have something like a thermostat. So a homeostat, like the thing that keeps temperature in your house, also a piece of physics, also could be rewired if you wanted to, but it avails you of something much more interesting. You can change the set point. And when you change the set point, that means you can walk away. And what can you do? You can trust the system to maintain the temperature, even though you're not there to micromanage it. So that ability to pursue goals, that's a tiny little, in the cybernetic sense, is a tiny little goal. It's a goal of keeping the temperature a certain way. And those kinds of homeostatic systems avail you as the engineer of a new way of relating to it. You don't have to rewire it when you want to change the temperature. You just change the set point. Then sort of further to the right of that are other systems that can do interesting kinds of learning. So maybe they can do habituation, maybe they can do associative learning, rewards and punishments. Humans, so the nice thing about those kind of systems is that even more so than with the thermostats, you don't need to know almost anything about how the system is made in order to control it. You just need to know what the rewards and punishments are. What are the currency? What does the system care about? And so humans have been doing this, of course, for like 10,000 years. They've been training animals knowing zero neuroscience. You don't need to know any neuroscience. What you need to know is how to motivate the system, and you need to know that it is in fact a learning system. You're not going to train a clock, but you might train certain kinds of living systems and increasingly certain kinds of non-living systems, right? There are plenty of non-living systems that will learn that way. So and then to the right of that, you have far more sophisticated systems that have metacognition and second order models of themselves and their world, and some of them have language like humans. And there you can do something even more interesting. You don't need rewards and punishments necessarily. You don't need to know how the system works. You can provide reasons for them to do things. And if you're clever and you are good and you are understanding how to communicate with the system, you can provide reasons for them to do things and then walk away, and then they will do all kinds of complicated things, and you don't have to micromanage any of it. So to me, it's all of this is really an engineering question about what level of sophistication does this type of mind offer when we relate to each other. So that's the continuum I have in mind. And you can place anything on that continuum, no matter what it's made of, whether it's alive or not.
So basically, let's say that if we walk on Earth and we just pick up random objects, it can be a human, it can be any other animal, maybe a microbe, or it can be a piece of rock. So we'll just look at like if this system is able to kind of, so this is what you mean by cognition, right? That it is able to perceive, so basically receive the signal and have memory and act on that memory. So these are the three things you would look for, or
I look for to the framework that I've put out focuses on the issue of being able to pursue goals. So a goal directed system. Now the next question is, well, with what kind of competency? So you could pursue goals maybe you're such a simple system that all you can do is go directly towards your goal. That's all you know how to do. So for example, magnets in the magnetic field or bowling balls in the gravitational field, they always go in a particular way. They're never going to do anything else. They're very easily prevented from reaching their goal by local maxima. So you put a little hill and they bump into it and that's it. They're not going to go around. They don't know how to do anything. They have very little competency in pursuing these goals. Then you have thermostats, which are much better at it, but they still have limitations because there's only a few things they know how to do. They don't set their own goals. Some other system has to set the goal for them. Then you have much more complex systems that have a lot of ingenuity. So if you've ever seen kids or advanced animals to solve problems, they will come up with all sorts of different ways to navigate their problem space that is not a direct line. In fact, they may have a degree of patience. They may know that it's better to step further away from their goal and then go around in some more complicated way to actually get closer to what they need. So the degree of competency in navigating your space, whether low or high, is the degree of intelligence. But the central thing that ties all of these things together is the ability to pursue goals. So goal directedness is what keeps all these agents on the same continuum. Now, one important thing you said is, you walk around and you pick stuff up. This is very important. A lot of people think that where something belongs on this spectrum can be decided a priori. So you're sitting here in your philosophical armchair and you look at a gene regulatory network, this is genes turning each other on and off, and you say, well, I can see it's very simple. It's a piece of physics. There's no magic here. It's going to be zero on the cognitive scale. I mean, there's nothing here. You can see, but actually, if you're willing to accept that this is an empirical question, you can't just have feelings about this stuff. You have to do experiments. You might find that, well, actually, gene regulatory networks have 6 different kinds of learning, as we found. And so the thing about all of this is that this is meant to be the reason. So my framework is called TAME. TAME stands for Technological Approach to Mind Everywhere. And the reason it's technological is that I don't believe these are philosophical issues. I believe these are empirical engineering issues. And if you want to know how cognitive something is, you have to test it. You have to make a claim, you have to test it. And if somebody else has a different claim that tests better, then they're correct, and so on. So all of these things we have to test. We can't make assumptions.
This is interesting. And I think also people should understand that how much in the science we have metaphors, and the fact that we are using living system, non-living system, those are metaphors, but now this is like the other framework that you're explaining, that this is kind of another metaphor, right?
Everything, well, I mean, that's true, but to keep in mind, I mean, everything is a metaphor. People say to me all the time, oh, well, it's just metaphorically that this thing has preferences and memories. I say, look, everything, how about the notion of a pathway, right? A biochemical pathway, or any of the things, any of the pretty color diagrams in this biochemistry book, they're all metaphors. Everything in science is a metaphor. And the only question is, how good is your metaphor in allowing you to make progress? That's it. There's no sort of objective truth and then some metaphors. It's all metaphors. All we can do is compare them, test them and compare them.
Yes, but the understanding in science, it evolves with certain laws and rules that we kind of test again and again, and we verify them. We know that, okay, this is this explains the system. And so how would then we'll apply evolution and selection on this continuum system?
Yeah, well, that's a very, very important set of questions. I have many things to say about it, but one of the issues is that I guess I'll just tell you two things. The fact that living things are nested, so subcellular chemical networks, cells, tissues, organs, whatever. That's a multi-scale competency architecture where each level has its own ability to solve problems and pursue goals in various spaces. We're talking the transcriptional space, physiological space, anatomical space, and then of course traditional behavioral space. The fact that these things all solve problems on their own and have some degrees of competency has massive implications for evolution. I'll give you one simple implication. One of the experiments that we did years ago was a study of what happens when you make tadpoles where the various craniofacial organs are in the wrong locations. Okay, so we tested. Actually, let's start even a step back. Tadpoles become frogs. And in order for tadpoles to become frogs, all their craniofacial organs have to rearrange, right? Because tadpole faces don't look like frog faces. So the traditional model of this was that, well, the genetics just somehow tells every piece of the face which direction to move and how far, and then you get from a tadpole to a frog. So that was the assumption. Now, we wanted to test that because I had a feeling that actually it would be more intelligent than that. And so how do you do that? You ask the question, okay, how much competency does it have to reach its goal? So what you do is you create, and we created these tadpoles, we call them Picasso tadpoles, where all of the organs are in the wrong place. The eye is off to the side, the jaws are on top of the head, everything is in the wrong place. And what we found was that actually, yeah, that process is much more intelligent. It actually, all those pieces will move around in novel paths, unnatural paths, and still get to where it's going. So what evolution has done is it hasn't given you a machine that does exactly the same thing and solves the problem in exactly the same way. What it gave you is an error minimization scheme. It's a machine that's able to minimize error, and it's able to get to the same goal from different starting positions, right? Different positions in anatomical morphospace, they all go to the same, basically the same frog face. That was William James's definition of intelligence, is the ability to get to the same goal by different means. So it has some degree of competency in navigating that space. It has some policies about how it gets there, and it's got some degree of intelligence. Now, here's the implication for evolution. So that's interesting. So evolution makes problem-solving machines like this. Well, here's the implication for evolution. Imagine that you're a tadpole and now you had a mutation, and that mutation, as most mutations have multiple effects, that mutation had a couple of side effects. One is that your mouth and your eyes are now in the wrong position, but it also has some other interesting changes elsewhere in the body. Now, under the standard scheme where it's a direct relationship between the genome, the genotype, and the phenotype, you would have a fitness of 0 because your eyes are off target and you can't see anything. Your mouth is off, so you can't eat. That's it. So you have a fitness of 0, and all the other consequences of that mutation are never explored. So this is the reason why the vast majority of mutations could be harmful, harmful mutations. That makes sense. As engineers, we know if you have a nice system and you make random changes to it, almost all the time it's going to make things worse. It doesn't make things better. Okay, but we're not dealing with that situation. What we're dealing with is a very interesting machine where the parts have competencies. So what that means is if your eye is out of whack, that's okay. It'll get back to it. It can still see. In fact, we've shown that animals with eyes on their tail can still see because the eyes will connect to the spinal cord instead of the brain. And those animals can see, if the animal has only the eye on its tail, no primary eyes, they can still see perfectly well. We can train them on visual cues. So if the mouth is in the wrong position, no problem. It'll get to where it needs to go. It can move and rearrange the face, and it will get to where it needs to go. So that gives evolution an incredible boost, because it means that now you can explore all the other consequences of that mutation. Maybe it does something really great somewhere else in the body, and it turns all kinds of harmful mutations into potentially neutral mutations. So it really puts evolution on steroids. The fact that you can depend on your parts to get their job done, even when things change. In fact, there are some amazing examples of this in biology that we can talk about. Anyway, so the first consequence is that it makes evolution much more efficient. It makes the power of evolvability goes through the roof. But the second is something else that's interesting can happen, which is that if your parts are competent enough to make up for certain kinds of genetic deficiencies, then here's what happens. Imagine you have two animals. They both look pretty good. They have good, adaptive behavior in the world. So here they come up for selection. Selection needs to choose which ones are the best ones. They look exactly the same. However, selection doesn't know whether they look the same because they have a fantastic genome, or they look the same because the genome was actually so-so, but all the parts kind of made up for it and everything is there. As soon as that happens, the power of evolution to make improvements by selecting great genomes goes down because you can't see the genome anymore. You are right. You're losing information. Your ability to judge and compare genomes isn't that great anymore. But what you can do is you can put more effort into improving the software, into improving the competency of the parts, right? And so I think that what happens during evolution is the competency of the parts make a ratchet for improved intelligence, because now it's very hard to make improvements by improving the hardware, but improving the software is really a powerful technique. And there's a great example of this. There's a great biological example of this, which is there's no, to my knowledge, there's no other model in biology that explains why this would be other than what I just told you. There are these flatworms called planaria. And these flatworms, they regenerate, and the way that the way they can reproduce is they tear themselves in half, and then each half regenerates, and now you've got two worms. Okay. So the interesting thing about that is that unlike us, when we have a mutation in our body, our children don't inherit that mutation, right? There's a separation of the germline. We clean it up during sexual reproduction. These planaria, because they reconstitute half the body each time from both sides, they accumulate somatic mutations. So any mutation that doesn't kill the cell makes it into the next generation and gets amplified as the cell, proliferates, right? So for 400 million years, these animals have accumulated these really all kinds of mutations. Their genome is an incredible mess. They're mixoploid. Every cell has a different number of chromosomes even. Okay, so the hardware description is all over the place, but they are champion regenerators. Their anatomy is 100% rock solid. Every single time they're cut, perfect shape. Now, this should be a scandal, right? We know nobody ever hears about this in developmental biology textbooks, but we all of our models predict exactly the opposite. Why would the animal with the most messy genome have the best anatomical control? That doesn't make sense, because you on the theory that your genome is what determines your anatomy. But what makes perfect sense is that this ratchet went all the way in this animal, that improving the genome became less and less important. And in fact, the pressure to clean up the genome became less and less important, because the competency of making a perfect worm, even though you're made up of a bunch of cells with different genetics and whatnot, that the algorithm got better and better. So I think those two things sort of positive feedback loop on each other. And that's just an example. So there are many other consequences, but those are two examples of evolution where the competency of the parts really changes the space that you're searching. Evolution maybe doesn't just search the space of all the kind of ways to have the hardware. It's more behavior shaping. It because you're not working, evolution, like bioengineers, doesn't work with a passive material. You're working with cells that used to be their own organisms. They have their own agendas. So what you're searching really is the space of possible communications, signals, inducements, rewards, punishments, all kinds of signaling. How are you going to get your material to do new things? That's what we as engineers face. That's what evolution faces. It's a very different task than searching the space of all possible configurations of your hardware.
This is great. So the major points which we should probably elaborate as well. So the first thing that you described is Picasso frogs. Then it's the genetics that we need to, and I mean, you kind of already mentioned a little bit planaria. That's another thing. And how basically once we mess up with parts of it, how do they change? And the fifth thing was regeneration. So let's start with the genetics itself. So when we are talking about genetics, so all the living forms that they have DNA or a sort of a blueprint, which is this kind of notion that that all of us kind of like everything is embedded in the genome, but that's not the case. This is what you are describing with the example of Picasso frogs, right?
Well, right. Let's unpack that a little bit. What does it mean for things to be embedded in the genome? First of all, we can read genomes now, and we know what's in the genome. It's protein sequences. That's what's in the genome. So now that's a description of the micro level hardware of the system. we know now, and I think in this case, the hardware software analogy is actually pretty useful here, because we know that, yeah, you can have a description of the hardware of the system, but then the things that that system is actually going to do are not just what was in the description of the hardware. They're actually, it involves all kinds of information from outside, for example, the laws of physics, the laws of computation, and so on, right? So let's imagine, I'll give you two simple examples, because this is important. People often talk about what's in the genome, where is shape encoded. So let's talk about this. You may have heard of a Galton board. So a Galton board, right, is you get this piece of wood. It's up here like this. You bang a bunch of nails into it vertically. You take a bucket of marbles and you dump the marbles into the top of this thing. They all sort of bang, 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 bang. They all do this. They come down and they make a lovely bell curve every single time. To make a bell curve. Now, you never know where an individual marble is going to end up, but overall they make this very nice bell curve. So you might ask the question, okay, that's a really cool morphology, that shape. Where is that shape encoded? You could study the wood, you could study the nails, you could study the distribution of the nails. It's not there. Where is it? Well, it's in the laws of physics and the laws of mathematics and calculus, and that's what gives you the bell curve. Now, here's another example. I could give you a specification of some transistors and some resistors and a few things like that, and you will make an AND gate. When you make an AND gate, the AND gate has a nice truth table that tells you how this thing works. And in fact, if you make a NAND gate, you can make all sorts of really complicated, computations, right? So now you say, well, where's that truth table? You didn't give me the truth table. What you gave me was a set of descriptions of transistors. So where does it come from? Well, it comes from the same place that the truths of mathematics and number theory and so on come from. I have no idea where they come from. The point is that we know perfectly well that for most devices, the most important thing that they do is not, in fact, in the description of how you built them. DNA is exactly the same way. So DNA, when you look at a genome, you don't see anything about the size, the shape, the symmetry type of an organism, whether it's regenerative, how many eyes it's going to have. You don't see any of that in the genome. What you see in the genome is a description of the micro level hardware that every cell gets to have, the proteins. Now, what evolution has done is to shape that hardware so that it gets to use certain gifts from physics and computation and so on. These affordances are like generic laws that you don't have to evolve specifically. So for example, if you had a triangle, you don't have to evolve 3 angles, you can only evolve 2, and you know what the third one has to be. Now, why is that? Well, it's a free gift from geometry. I don't know where it lives wherever mathematics lives. So the point is that in that sense, the genome does not directly encode the shape. It doesn't encode a blueprint of any kind. It encodes hardware that does something amazing. What that hardware does is it builds a machine that solves problems. One of the problems that it solves is how to self-assemble from a particular starting state. But most of them actually solve other problems too, other problems which are, for example, well, how do you repair yourself if you're damaged? How do you build yourself from a different starting state? What happens if your cells are much larger than they need to be? For example, in the newt, you can make the early cells gigantic and you still get a perfectly normal sized newt because fewer cells do what they need to do. And in fact, some cells will like wrap around themselves to make a particular kidney tubule and so on, whereas normally it takes 8 to 10 cells to do it. They'll choose different molecular mechanisms to get to the same goal. So the hardware is amazing. It does all kinds of things, but like as we know, like with computational and other kinds of machines, it's not in the description of the machine. It's the machine using the laws of physics and cybernetics and computation and so on.
Yeah, the question would be that so first of all, what would be the software then? And if the software can change the hardware?
Yeah, the software is the physiology. The software is the computations being done by bioelectrics, biochemistry, biomechanical forces. All of the physiology that happens is the software that ends up doing things like, well, if you start off in the wrong position, it will fix you. If you're a salamander and you lost your arm, it will keep growing the new arm until it stops. That's the software. That's now can the software change the hardware? In some sense, yes, because you most certainly change the large scale hardware because that's what the software does. It builds the organism, it remodels the organism. It's what makes a caterpillar become a butterfly. So, I mean, radical changes of the hardware. not only that, but it also potentially can change the hardware at the lower level. So certain cells will die, other cells will come, different genes will be expressed, the cells will move around. Maybe at some point there are changes even of the genetic material itself. I'm not sure. There are cases have been made for bacteria for transgenerational inheritance in other animals, maybe. So yeah, but that's the idea. The software is the physiology. It's the layer between the genome and the anatomical structure and function that provides the intelligence of the system. It's that layer in between.
And that would also explain that these systems are not deterministic in a way, right? That there is plasticity there.
Well, yes. So from the biological scale, absolutely, there's incredible amounts of plasticity, and we can talk about some amazing examples of living things being interoperable and forming. They can make coherent organisms as chimeras between different DNA and different cells and different organs with inorganic materials. People grow things on crazy material scaffolds and everything. I mean, amazing plasticity. At the, if you want to be sort of really pedantic about it, at the micro level, they are deterministic in the sense that they're really, everything follows the laws of physics and chemistry. It's not magic. There are laws of physics and chemistry. And so you could argue, I mean, there's the whole quantum question, but you could argue that at the micro scale, they're absolutely deterministic. But that doesn't mean that at the scale where it's really important, which is the larger scale of anatomy and physiology, yeah, they're not deterministic. They're very there's massive plasticity.
Yeah. So now, okay, let's see again. So now, if we start from a single bacterial cell, which is like the simplest living forms, again, with certain blueprint, with certain information, as you said, or hardware, right? Where I think the hardware would be, or can we say that the hardware would be more dominant in a way? And then once we go into more complex forms where, of course, the number of cells increases, there is, of course, the issue of multi-cellularity. And once that case comes, it's more of the software-driven phenomena.
Maybe that's a hypothesis that the problem with being able to be certain about that is that all claims like that are relative to an observer. So you and I look at a system and we say, oh, well, this isn't doing anything too sophisticated. I think it's mostly pretty hardwired, right? The problem is that when we do that, we are taking an IQ test ourselves. So we are saying, how smart am I in being able to notice what this thing is actually doing? Because you have to do three things. You have to pick a problem space. You have to make a guess as to what the goals of that system are. And you have to make a guess as to what the competencies are. That's a lot of guessing. And then you have to do experiments. Now, think about this, that as we grow up, all of our sense organs basically point outwards. And so we are very good at recognizing intelligence and plasticity in medium-sized objects moving at medium speeds, in our lifetime, that kind of thing. And so when we see animals and birds and dogs and humans doing interesting things, it's very easy for us to detect agency and to know that they're different from bowling balls and so on. Imagine if we had the way that we have vision. Imagine that we had the same kind of built-in direct perception of your blood chemistry and all the things that your liver and your pancreas are doing to keep it, maintain the chemistry. I think in that case, we would be quite good at detecting intelligence in physiological space, not in three-dimensional space, but in the space of physiological states, because you could see everything, you could feel everything that was going on. And then you would see what your organs are doing and you say, wow, that's really clever. Look at what it did. It anticipated that every day at noon I have this like heavy lunch, and it anticipates and it does interesting things. So all of this, we have to be humble about these things to know that we may not know what it's doing. So for example, bacteria, we may think they're hardwired, but I would bet, and I'm not a bacteriologist, I'm sure other people could tell you better, but I would guess that in metabolic space, these bacteria are incredibly plastic. And I think that we would find out that there's a huge amount of software going on at the metabolic sort of chemical space that the bacteria are dealing with. They don't, they don't do terribly much in other spaces, but I bet in that space, I bet they're very smart. And so, but these are all conjectures, right? So people do experiments, and lots of people study this stuff, and we could get real data on this, and then we would know for sure. Until somebody else came along with a better model, of course.
Yeah, so the other thing that you were describing is the Picasso frog. So what exactly you do there? So do you change the genetic material?
There's a few ways to do it. You could do it surgically, you could do it bioelectrically, you could do it genetically. We've shown example, and so what you do is you make a tadpole that has the wrong configuration of the face, and then you find out that the system actually knows perfectly well what a correct frog face looks like. How do you know that it knows? Because it's able to reach that despite deformations. So that's what makes this engineering as opposed to philosophy. When I say it knows, I'm not using some sort of crazy metaphor where we can argue philosophically whether it knows. It's very testable. We know that it knows because it will always get back to that same spot, even though you put it in different places. And in fact, we can even begin to read that memory. You say, how does it know what a correct face it looks like? We've actually developed techniques where we can see that memory. We now know how they know. So, the thing about it is that if you didn't do the experiment, you would never have found this. Like I said, we don't know the sophistication of these systems until we try it. We have an example where we can use software to fix certain kinds of hardware problems, not all of them. If you're missing some sort of enzyme that you need for metabolism, bioelectrics is not going to help with that. But we have an example where we looked at the tadpole brain, and there are a bunch of ways to disrupt it. So there are chemicals that will disrupt it. There are genetic mutations, for example, in a gene called Notch, which is really important neurogenesis gene. So what we showed is that that the way that the brain knows how big to be and what shape to be is a particular bioelectrical memory in the cells before there's any brain, in the cells that are going to be brain later, that tell them what the brain should be like. And what you can do is, even though they're impacted by nicotine, alcohol, or mutations, genetic mutations or changes of the hardware, even after that, you can treat them with a particular set of drugs that opens and closes electrical ion channels that restores the correct bioelectrical pattern. So we use the computational model to say, this is what the correct pattern is. How do we manipulate the voltage to get back to the correct pattern? So this wasn't a screen, it wasn't a guess, there's a model that tells you how to do it. So we did that. And sure enough, you get a normal brain with normal structure, normal gene expression, and their IQ goes back to normal. So they actually regain their learning rates. So that tells you that at least for some cases, you can repair genetic hardware defects in software, but that's going to be on a case-by-case basis when that's possible.
Yeah, let's try to put it again in the same framework that we were discussing. So the genomes, they code for RNA, proteins, and these proteins are the basically building blocks doing different things. Of course, there are also RNAs. And then so these things are again packed in the cells, which can, of course, communicate with each other. And so basically what you're doing is that we can, and all of us, all the multicellular organisms, that they are built because of the communication between these cells, right? And these communications can be changed genetically. So we change the hardware, but we can also change these proteins, or there can be some ions. And these ions are, for example, for neurons, we know that these neurons fire, and that's what we call action potential and these kind of technical terms. But this is how this has been known for years, that okay, this is what happens in the brain. But now this is another mechanism that you guys are describing, which is called bioelectricity, where the similar pattern is basically noticed in all the cells, all the somatic or any cell in the body.
Yeah, so let's just be clear. Well, let's start with the brain, though, the kind of familiar scenario. It's not that ions are somehow magic for cognition. Okay, there's nothing particularly special about ions, except that electrical networks happen to be really good at processing information, at making feedback loops, at storing memories. And that's because they take advantage of certain laws of physics and mathematics. So the brain uses that modality as the cognitive glue, right? We are a collective intelligence. You and I are bags of neurons, and something enables those neurons to cooperate to have an emergent set of goals and behaviors and memories and preferences that don't belong to any individual cell. They belong to this weird emergent entity known as a human. So it happens in the case of on Earth that evolution chose electrical networks to be able to do this. Now, the thing is that this system, evolution discovered this long before there were any brains. So this is a trick that evolution picked up around the time of bacterial biofilms. So there's some beautiful work out of, for example, UCSD, where they show bioelectrical kinds of dynamics in bacterial biofilms, in bacterial mats. So this is a very ancient thing. All cells do this. What evolution seems to have done is it took a set of mechanisms that were used to solve problems in morphospace, so in anatomical space. So problems like which cell goes where, how many eyes, where does the head go, where does the tail go, these kinds of problems, and took that exact same set of mechanisms and just pivoted them onto a different space, the three-dimensional space. So now, instead of moving the configuration of the body through anatomical space, now it moves your whole body through three-dimensional space by activating muscles instead of activating other cells and gene expressions and so on. It's the same trick. It's an ancient trick. Every cell group in your body does it. And it's not because, and again, it's not because bioelectricity is somehow magically, cognitive. It's simply that that's a great way to make use of a lot of computational laws. Which it's the same reason we make computers this way. It's exactly the same.
Yeah, but then what will be the difference between bioelectricity and any other kind of signaling?
In theoretical terms, you can do anything that you can do with bioelectricity potentially could be done with other forces. So probably somewhere there's some planet where instead of bioelectricity, something else is used for the same purpose. But here on Earth, the immediate benefits of bioelectricity are many. It's, electrical networks are, it's not an accident that brains use them, computer technology uses them. It's just it's very convenient. I give you a simple example. There are ion channels that produce voltage, but these ion channels themselves are voltage gated. So what you really have is a voltage gated ion conductance. It's an electrical gate whose open or closed state depends on the voltage, which means what happened before. So that automatically gives you a bit of historicity, right? So it's got this like dependence on past conditions, which automatically makes it be two things. It makes it a synapse, meaning a communication channel that has plastic enough to depend on what happened last time. And so that's a bit of memory. And it enables you to make transistors, because that's what transistors are, voltage gated current conductances. So as soon as you have that, we know what you can do when you have transistors. You can compute anything. And so that's why bioelectricity is so interesting. Now, could you do this with other things? Of course you can. And but this is one reason why every once in a while in nature there'll be a nice paper on some sort of diffusion powered logic gate or something like this. That's why it's in nature, is because it's so damn hard. It's really hard to do that kind of thing with diffusion, with chemical signals, biomechanical signals. Not that it can't be done, but it's just really hard.
But this voltage gated ion channels, they basically deal with the simple salt that we eat, or like sodium, potassium, these ions, right?
Yeah, the cellular resting potential, the electrical properties of each cell is set by the balance of sodium, potassium, chloride, protons, and some other things. And so, but again, not magic, just a cool piece of physics that allows evolution to harness certain laws of computation.
And so the Picasso frogs, now back to this point. So in those frogs, do you change the bioelectrical signals then, or what exactly?
We do, yeah. So for many purposes. So for example, one thing that you can do is you can say, how does the face know where to put an eye? And so you look at so we developed a technique using voltage sensitive fluorescent dyes that show you where all the voltages are distributed. And when you look at the early region of the skin that's going to become the face, you can actually see some patterns in that voltage that tell the cell, here's where the eye is going to go, here's where the mouth is going to go, and so on. What you can do is you can reproduce those patterns somewhere else in the embryo. And you can do that by opening and closing ion channels with drugs and with other things somewhere else. When you do that, you reproduce that bioelectric state somewhere else. Those cells will, if you do that for the eye pattern, they will make an eye. And we've made frogs that have eyes made out of gut tissue, on the eyes on their tail, anywhere you want. We can make eyes, legs, brains, a few other things. So what it ends up being is a kind of subroutine call, because we don't have to micromanage, again, taking advantage of the intelligence of the system. We don't have to micromanage it. We don't have to tell it how to make an eye or where all the different cell types go that make an eye. It's very complex. We don't know how to do it. What we do know is a high level signal that tells the system, make an eye right here, right? It's basically a very simple subroutine call. And then the cells already know how to do it. And then they do it and then they stop. So again, this is a system that isn't all the way on the left of that continuum where I would have to guide I'd have to 3D print every, people want to do this. They want to sort of micromanage the positioning of all the stem cells and all the connections, and the different genes go up and down. They want to control all of that. That's okay, but that's incredibly hard for a complex system. It's much easier to realize that your system is actually quite a bit further down on that continuum, and it understands goal states and it understands modularity. And you can give it signals that say, build an eye here, and then you walk away. So we've done that kind of a thing. We've done another thing where frogs, unlike salamanders, do not regenerate their legs. So we showed that you can make them regenerate their legs by putting a particular stimulus on the amputation wound for 1 day, so 24 hours. Then for the next year and a half, they grow the leg. We don't touch it during that moment because we're not micromanaging the process. We're not trying to control it. We are trying to communicate with the decision-making process during that first 24 hours that says, are we going to scar or are we going to regenerate? And what are we going to regenerate? All that decision-making machinery acts at the beginning. Once you've communicated with it, you can take your hands off and let it do what it needs to do. That's what's important about all these things, is that it tells the engineer how to effectively control the system.
Yeah, the regeneration part is very interesting. But before that, let's also cover a few points here. So the first point would be the expression of the voltage gate channels, how much that changes with the different parts. The second thing is that when we are talking about bioelectricity, what is changing in the different parts of the embryo or organism in general? Is it the concentration of the ions, which I think it would be fairly the same, but or
no,
it's just
the ions, yeah, absolutely. Voltage is set by the balance of between inside and outside of the cell of the various ions. So the ions go in and out of the various cells. The total is more or less the same. If you just grind up the tissue and ask how much total potassium, yeah, it's about the same. But the real question is, where is it? Is it inside the cell, outside the cell? That's the really key. And that's the same thing for neurons and action potentials. Because the question is, where are these ions, in or out? So all of that changes during development in very, very specific ways. And that results in a set of bioelectrical distributions. It doesn't matter what any one cell voltage is. It's not one cell. This is not a cellular code. This is a tissue or organ level code. So you look down and you see these different bioelectrical distributions, and different stages are pulled by the fact that that distribution changes, and that distribution is used to store the memory against which the homeostatic agent measures. So for example, so the key to all of this is that any homeostatic agent has to have a memory of the set point, right? If you're going to be a thermostat, you need to remember somewhere what the correct temperature range is. Same thing here. If you're going to rebuild a face, if you're going to remodel a face, rebuild a leg, all of these things, you need to remember what the correct pattern is. How does it remember? Well, it turns out that the way it remembers is to store a bioelectrical pattern, which is a rough description of what it's looking for. And the error, the delta between the current pattern and the correct pattern, is what drives change. It drives remodeling. It's a kind of stress there's a stress reduction loop that tries to reduce that that error. And so that bioelectrical pattern keeps changing, and as it changes, it pulls the gene expression and the anatomy with it, because they will keep changing to try to implement what the bioelectric pattern is doing, try to reduce that delta. So you go from stage to stage to stage, and eventually you flatten out as a mature organism.
But does this mean that this bioelectric pattern would be different in, for example, liver? It would be different for kidneys, or it'll be different for eyes?
Yeah, yes, it's different for different structures and different organs. The full story of what all the patterns are for all the different shapes, we don't know all of them. That's very much ongoing research. We call that cracking the bioelectric code. We need to understand the mapping. We need to understand why specific patterns make specific shapes. And so that's very much still under investigation. We know it for some systems. We know it for a few things, but we don't know it for everything. So for example, liver, I have no idea what a correct bioelectrical pattern is for the liver, but we know what it is for the face. We know what it is for the limb. We know what it is for the brain. We have some information.
And what is the meaning of this bioelectric pattern? Like what exactly you're looking at? It's the ions, or also the type of channels?
Neither. It's not any of that. That's all hardware. We are looking at software state. So what we're looking at is voltage. So we are specifically looking at the distribution of voltages across a sheet of cells, right? And the cool thing about it is that the voltage is a function of many different ions. And what we have shown repeatedly in all of these papers is that you can get the same effect using sodium, potassium, chloride. It doesn't matter what the ion is, as long as you get the voltage correct. So you can use any of them, but you have to pull them in or out using the optogenetics, so light gated channels, or drugs to open and close the channels, or put in new channels, whatever. The point is, you just have to get the voltage pattern right. So the system does not track the microstates of which ion did it. It tracks the resulting voltage. The same way that, and this is something computer science figured out a really long time ago, that when you are programming at a higher level, you don't need to know where the voltages are in your computer, right? When you on your computer, when you change from Photoshop to Microsoft Word, you don't get out your soldering iron and start rewiring, and you don't have to keep track of which piece of copper has your currents. You don't need to know any of that, because the system exposes a higher level interface. You can program it with experiences, with stimuli, and you know something about how the thing works. That's what we're looking for. We're looking for the ability to read and write these voltage states, and that way we can basically avoid having to worry about all the microstates. You know, right now in biology, I mean, you've seen those pictures of what programming looked like in the 40s and 50s, right? You sit there in front of this giant thing and you pull wires back and forth, right? How much fun was that, programming that way? And that's where we are in biology and medicine today. We are all about the hardware. CRISPR, genomic editing, stem cell biology, pathways remodeling, gene regulatory networks. Okay, great, but all at the level of the hardware. We have to move beyond that, because some problems are not easily solved that way. Okay? And we can see computer science already took that journey for us, so we can see how that works.
Yes, but then of course in the future we'll be going at like more specific level to kind of find out those switches which turns on and off certain switches, right? Like the certain.
Yeah, thinking about it from the perspective of switches is okay, but it kind of misses the point. It's sort of like if I gave you a computer that plays chess, and you wanted to study it, well, you didn't know what it was, but you wanted to study it, you could get a really great accounting of all the materials, where all the copper is, where all the silicon is. You could study all the switches. You could measure all the magnetic fields generated by the electricity. You could get a great, you might even, if you had, sort of a lifetime of many universes, you might even get to the point where you could predict what the next electrical step is going to be. You would be none the wiser about the game of chess. You would not know anything about the algorithm. You wouldn't know what it is, what it means to protect the king and dominate the center of the board, or any of this other stuff. It's just not available at that lower level in any obvious way. So I think for regenerative medicine, it's the same. We can study all these micro switches, or we can ask, how does the system make decisions? What is it measuring? What is it remembering? Why does it decide this or that? And what are the signals that are going to make it, sort of push it into the actions that we like in morphospace, building certain organs and whatever. So that's what I'm excited about. I mean, working out the switches is fine, but I want to communicate with the system at a higher level.
Yeah, and it's definitely exciting. And since you already mentioned about regeneration, let's talk about two-headed planaria.
Yeah, well, the thing about the two-headed planaria, it goes back to our question about what information is in the genome and how does it know what to build. Now, let's start off. Let's just start off by reminding ourselves of some knowledge gaps here. Let's do a thought experiment. I have two different species of planaria. One have a round head and one have a flat head. They have some stem cells, and each planarian, when you cut off the head, they build a head of the correct shape, and then they stop. That's what planaria do. Now I have a thought experiment. I'm going to take my round-headed planaria. I'm going to take out some stem cells, and I'm going to inject them into the body of the flathead planaria. I'm going to let them get comfortable and move around and settle in, and then I'm going to cut the head off. What head shape are we going to get? Is one head shape going to be dominant to the other? Are they going to get an in-between kind of intermediate shape? Or is the shape never going to stop because neither set of cells is ever happy about what kind of shape we've made, right? Nobody can agree on what the shape should be. Now, we have some beautiful papers in Nature and Science and so on studying stem cells in planaria, plenty of information. We have the genomes. Why can't we answer this question? Why do we have zero models that make a prediction on this thing? Because we do not understand. We understand how the hardware works. We do not understand the software. We do not understand how collectives of cells make decisions about shape. And until we understand that, we can't answer questions like that. So we wanted to ask a simple question years ago. Here are these planaria. You cut their head off. They make a perfect head, always the right number of heads at the right position. How do they know? How do these cells know how many heads there should be and where these heads should be placed? And so we looked, I made a hypothesis that in retrospect, it doesn't say, I mean, at the time, everybody said it was nuts, but thinking about it in retrospect, it shouldn't be that surprising. How does the brain store memories? Electrically. Okay, so we used a voltage dye to look at the planaria, and we noticed something interesting, that there was a bioelectrical pattern that looked like it was storing the information to have one head. That's what it looked like. And so then we said, okay, very simple. What happens if we artificially change that bioelectrical state to be a pattern that looks like it has two heads? We don't change the DNA. The hardware is wild type. We don't change we don't edit the genome. We don't do anything like that. We don't put in new channels, even nothing. We just use a drug to control the bioelectrics so that it now says two heads. And now you cut it. Guess what? It makes the fragments make two-headed worms. So that's interesting. So you learn from that the instructive information about where the heads should go lives in this bioelectrical circuit. Now, we learned something else when we took the two-headed animals and we recut them again into pieces. No more manipulation of any kind, no more drugs, nothing, just recut those two-headed animals. Guess what? They keep making two-headed animals. So that tells you that the circuit has a memory. Once you reset it from one-headed to two, now it stays, it's permanent, and we know how to set it back to one-headed. So that's pretty interesting. That suggests that the information on how many heads a planarian should have is, strictly speaking, not in the genome. Here's what the genome does. The genome creates a piece of hardware that by default has a software state that says one head. But that hardware is actually very clever. It can not only do that, but if you change it, it will also keep a new pattern. So we're familiar with this. If I give you the description, the parts list to a calculator, you put them together according to the description. By the way, my description says nothing about numbers. It says nothing about mathematics. It's just a description of a bunch of electronic parts. So you put these parts together, you turn on the juice. What do you see on the LCD? You see zero. You see a particular pattern, zero, every single time. Every time you turn it on, you see zero. Wow, that's amazing. It's reliable. It's much like embryonic development. It's robust, zero. But then you find out something interesting. Without having to rewire it, I can give it inputs, stimuli. I can press the buttons, And instead of 0, it'll say something else. And in fact, it has a memory and it'll say something else and it'll keep it, even though I haven't changed the hardware at all. There's no more mystery in these planaria really than there is in this example. The genome gives you the circuit, and the circuit is amazing in that it does two things. It has a default reliable behavior, but it's also reprogrammable, right? And that's what you've got. You've got biological tissue is reprogrammable. It has a default behavior, but you can communicate with it at a higher level and say, I don't say where the stem cells go. I don't say how many eyes you're supposed to have. I don't, none of that. I say 2 heads. It's a very high level communication. And then the cells build.
So how do you change the bioelectric signals? And in these two-headed planarias, did bioelectric signal change?
Yeah. So the way you induce the change is the way you make the two-headed planaria is by changing the pattern. And the way you change the pattern is you use a computer model to tell you which ion channels can be opened or closed to give you the right voltage results. It's not obvious because it's quite a complex circuit. So you can use a computer model, and the computer model will tell you that if I open this chloride channel for a while and then turn off this potassium channel, then you'll get this particular voltage. So that's the future of this field is development of these kind of machine learning tools that will be able to infer interventions and help you communicate with the system.
And we already know most of these voltage gate channels that and this is what then we can model, right? Or
yeah, and in fact, you know what all the channels are because you can have a transcriptional profile of the tissue, so you know what all the channels are. Those are your control knobs. Now the next question is, right, so you know what all the buttons are on your device. Now the next question is, which ones do you hit? And that's the more interesting question. That's what you need the machine learning part is to how do you communicate with that device?
Yeah, and you've been using this system already to create the famous Xenobots and also Xenobots.
Well, the Xenobots have not yet are not yet bioelectric. I mean, of course, we're doing that now, but the 3 papers that exist are not bioelectric in any way. So what the 3 existing papers on Xenobots are doing is showing the plasticity of the tissue. It shows you that. So for example, regular development is so reliable that it kind of it's misleading in many ways because it makes you think that this is what the cells want to do. This is the only way they can do it. So for example, if you ask, so two ways in which it's misleading. One is that you have a tadpole and it's covered by skin, and its skin is this like 2-dimensional passive layer on the outside of the embryo, keeping out the bacteria. And you say, okay, I can say 2 things. I can say that evolution, if I ask why does the tadpole look a certain way, why does it have certain behaviors, certain distribution of features, because for millions of years evolution selected for a very specific thing, right? And everybody else died off, so now this is what you have. And if I ask what do the cells want to do, well, they want to be this like passive 2-dimensional layer on the outside of the embryo. So again, you do experiments, and the experiment is you don't add anything. We didn't put in any new nanomaterials or new synthetic biology circuits or nothing like that. We engineered by releasing constraints. So what we did was we took away the other cells. We say, here are the skin cells. Let's remove them, liberate them from the native environment where they're normally told what to do by these other cells, and see what they want to do. Well, there are many things they could have done. They could have died. They could have crawled away from each other. They could have made a two-dimensional flat layer like cell culture. Instead, what they do is they show you what they really want to do when they're not bullied by the other cells, when they're not when they're not behavior shaped to be a skin layer. They form this 3-dimensional, very active creature known as a Xenobot. It swims around on its own. It has behaviors. It's able to make copies of itself by assembling other pieces of skin together. It does all kinds of interesting things. And that's what these cells normally do. How come that's what they want to do? How come they don't normally do that? Because what evolution has done is figure out how to make the other cells force these cells to do that. Right. So again, as I was saying at the beginning, evolution is all about behavior shaping. It's not about controlling the hardware. The hardware does what it wants to do, but the behavior shaping, the signals are what give you the outcomes. So yeah, you find out about the plasticity, that evolution, in fact, does not give you machines that solve a specific problem. They give you a more general problem solving device. And that it actually, if you ask where do the properties of the Xenobot come from, it's hard to say it was selection. There's never been any Xenobots. There's never been any selection to be a good Xenobot. There's never been pressure to be a Xenobot. Why are they Xenobots? Because there's creativity, because the intelligence, or the collective intelligence of these cells is able to come up with new solutions to problems. And in this new configuration, it figures out a way to be a coherent organism.
Yeah, so basically you just take these skin cells and they form into kind of small organoid kind of things. That the question would be, how do they control their size? Do they have like different sizes in general? So at the end, it is again the same thing, that it is about the cell to cell interaction, which or the cellular communication, which changes, and they now kind of form this Xenobots, right?
Yeah, I mean, the size, it depends. You can somewhat control it by putting different numbers of cells into a pile. But when the Xenobots make other Xenobots, when they make copies of themselves, they will make certain sizes by corralling a certain number of cells together. How that works, we're not sure yet. Yeah, and then like with everything else, we as engineers have to figure out what are the stimuli that we can give these things to get them to make new shapes. I mean, with some of that in the papers, you can see we use AI, we use machine learning to come up with ways to sculpt these things. It's just the very first step. There's going to be a million other ways to do it, but the very first step, which is very simple with sculpting, that we can use the machine learning to predict ways to sculpt it in ways that, and all the machine learning was done by Josh Bongard's lab at University of Vermont. They make these tools that allow us to search the space of possible interventions and tell us how to relate to the system. How do we control it?
Fascinating. And so about the regeneration again, and since we are again talking about this stem cells and most likely the somatic cells, right? So what is the difference? So is it possible that the regeneration is most likely close to or is dependent on stem cells itself? Now, of course, the question would be, what does this mean to have stemness in the cells?
I mean, well, it's known that the stem cells in planaria produce all of the tissues. So in order to have new cells, you need stem cells. So of course, yeah, it absolutely is dependent on the stem cells. But that's like saying, if somebody says, hey, this computer is able to play chess, and I say, yeah, that's because of the great silicon that's in there. I mean, kind of, that's true, right? It wouldn't happen without, or you say, well, that's because the power's on. Yeah, kind of true, but misses the point. So you certainly need the stem cells. You certainly need the stem cells to be making other cell types. But teratomas, they're tumors that have skin and teeth and hair and bone and all this stuff, and they're a terrible mess. It's a tumor, not an organism. The stem cells did the right thing. They all made their tissues. They made hair follicles and skin cells and bone cells. The stem cell biology is fine, but it's not enough to solve these problems.
Yes, but I think this explanation kind of explained that why. So let's say if someone cuts our hand, it's not like it'll regenerate, right? And the reason would be whether the stem cells and their communication. I mean, just because we don't have the stem cells or as many stem cells.
I'm not sure that's true at all. Frogs are like humans. They don't have a lot of stem cells the way that planaria do. They do tissue renewal. So skin makes skin, bone makes bone, muscle makes muscle, and so on. And we were able to make frogs regenerate their legs. We didn't do particularly anything to the stem cells. I'm not sure. Deer regenerate their antlers. So it's a large adult mammal that grows 1.5 centimeters of new bone every day when they're regenerating. So I'm not sure. I'm not sure to what extent actually stem cells are going to be a huge part of this, but maybe. Certainly, maybe certainly, even if they're an important requirement, they're not sufficient. They may be necessary, but they're certainly not sufficient.
So that means the major driver would be something like bioelectricity.
I think so, but the thing I'm sure of is that the major driver is going to be an informational process, which maybe is driven by bioelectricity. I would put my money on that, but that remains to be seen. But the thing for sure is going to be the driver is the decision by the cellular collective of what are we going to build. That's the first step. If you don't have that, nothing works and nothing's going to happen. And that may be fundamentally a bioelectric process, or it may include many other things.
So let's talk about the role of your work in health, in medicine, and in general, like how better we can study biological system.
Well, and I have to do a disclaimer here because I have a company that David Kaplan and I co-founded called the Morphoceuticals Inc. So we are pursuing this kind of thing. But to me, the biggest barrier to really transformative regenerative medicine is the universal assumption that living things are best controlled at the micro hardware level, that we should be looking at pathways, rewiring pathways, individual genes, genome editing, that kind of stuff. There will be low-hanging fruit that are addressable that way, I'm sure. But the general power of regenerative medicine is not going to be achieved that way, I don't think. I think what we do need to do is to figure out what the innate competencies of cells and tissues are. We need to understand how to communicate with them, and we need to exploit the intelligence of cellular collectives to call up outcomes that are too complex for us to micromanage, like growing back organs and so on.
Perfect. And what are the major questions coming next from your side?
Well, my group works on a number of interesting things. So we have projects in cancer, on repairing birth defects, on regeneration, of course. We're doing some things on machine learning, creating kind of robot scientist platforms that help human scientists make hypotheses and discover better ways to communicate with electrical and chemical interfaces in regenerative medicine. And of course, developing further these conceptual ideas around what is agency and how do we best understand other minds.
Yeah, this is so exciting, or at least I am really excited about it. So thank you so much for accepting the invitation, sharing your impressive work. And I think it has the ability to transform our thinking about biology. Thank you so much.
Thank you so much. Yeah, thanks for the conversation. I appreciate it.
Yeah.
Thanks.